Good morning, everybody. It's a great pleasure to be here because I think it's so important that we put our stuff, which we do in research, and really bring it to you, the persons who really count. And if you go to the main conference, it's very complex what we hear there. And I just told some of you this morning that even for me, it's hard to follow the talks of the other ones, of the other colleagues. So what I would like to do is give you an overview of the two main techniques which have been presented at this conference on last Wednesday, on the afternoon session last Wednesday. There were mainly two techniques which have been used there, which are of high importance in genetics and which really do revolutionize the field of ataxias, especially of rare ataxias. So I'm going to focus on recessive ataxias, novel genes, genetic mechanism, and diagnostic challenges. What is the problem we have in rare ataxias, or especially in recessive ataxias? The problem is that we have an awful long list of genes, a high list of numbers. There are more than 120 ataxia genes have been described so far. And if this is a series from our center in Tübingen, we have collected more than 200 patients with early onset ataxia starting below age 30, and we have searched could we make a diagnosis in them or not. And what you see here is that a quarter of them, 25% of them, do have Friedreich ataxia. So Friedreich ataxia is by far the most common recessive ataxia. That's pretty easy. We clinicians are quite good at diagnosing Friedreich ataxia. But the problem starts right beyond. Already if you go here, there is an awful lot of small, tiny fractions of rare ataxias. And almost nobody of us is able to diagnose this clinically and diagnose all those rare ataxia just by looking at the patients. And also we cannot sequence any single gene one by one. This would take us forever and cost an awful lot of money. So how do we deal with the fact there's such a large number of rare genes? And there's a second problem. 50% of our patients we can still not solve with the conventional methods. So there are two problems. One problem, many very rare ataxias and a lot of genes. Second problem, even if we sequence all the known genes, still up to 50% remain unexplained. So two problems, and what are the genetic solutions to those two problems? And I'm going to present them here. The first solution is that you use a so-called panel approach. A panel approach, I will discuss this in a minute, is an approach where you sequence all genes at once. Rather than going one by one, you just shoot all of them at once. And then you just cover all of them. How do we try to solve this problem? With a large share of unexplained ataxias, we do a so-called exome. I will explain this in a minute. But why is it important? Why is it important to do a panel or do an exome and find the genetic diagnosis? Why do we have to care at all about the genetic diagnosis? We could just say, oh, come on, whatever this stupid gene name is, we are maybe even tired of the stupid gene name, so why should we care about this? Only if you have a genetic diagnosis, you as a physician can give good information to you as a patient, can good information about the prognosis. And having a genetic diagnosis is the key to treatment. The future will look like this. We will not have an ataxia drug, which will work for all ataxias alike. We're going to have an individualized treatment for each or a few share of ataxias. So we will treat Friedreich ataxia completely different to SCA3, completely different to AT. And in order to enter those treatment studies, you need to know your genetic diagnosis. So we cannot live with the fact that 50% remain unexplained. So how do these approaches work, the panel approach and the exome approach? Let me describe this to you by comparing it to how we have done genetic sequencing up to 2010. Up to 2010, we have called a technique so-called Sanger sequencing. What is Sanger sequencing? Sanger sequencing is sequencing a gene, just a single gene. You focus on one gene. Let's say I want to sequence the RTM, the Lewy Body ataxia telangiectasia gene, and I sequence this gene. And if I'm done with this, I go to the next gene. And very often in the past, it looked like this. You have a patient where you suspect Friedreich ataxia. If it's negative, you go for the next gene, for example, the AT gene. If it's negative, you say, okay, it might be other early onset ataxia, it might be AOA2. If this is negative, you go for another, maybe Arsax. And this is how we have done genetic workup of our patients in the past, gene by gene by gene by gene. And if you go down this way, for example, those 4 genes cost 10 or maybe even 15,000 euro, and you might still not have the diagnosis. And it might cost more than 12 months to do this, and you might still not have the diagnosis. So up to 2010, this was our daily life, and this was frustrating for all of us. And now what are the advantages of the new approaches? The first approach, panel approach, or more elaborated targeted enrichment, this targets all known ataxia genes. For example, all known 120 ataxia genes. So rather than just shooting one arrow on the single gene you suspect, you shoot a whole range of arrows and just try to cover all ataxia genes. And this is pictured up here. So this is just a long list just to give you an impression of the power of the approach, that you can really target all ataxia genes at once and shoot all of them at once. And the advantages are here, right here. This just costs 3,500 euros. So even though you have 120 genes, it costs much, much less, for example, than this approach, which just covers four genes. And it's not only much more cost efficient, it's also much more time efficient. This just uses 3 months, maybe up to 6 months, whereas here we have said this takes more than 12 months. So much more time efficient, much more cost efficient, and you cover all ataxia genes, just a single one. And just thus increasing the likelihood that you can make a genetic diagnosis in your rare ataxia syndrome. So this is a panel approach. What is the exome approach? The exome approach is you're not shooting a single arrow, you're just not shooting 120 arrows, but you're shooting 20,000 arrows, meaning you cover all genes we humans have, not only the ataxia genes, but all genes, even those we are not yet aware of that they're anyhow linked to ataxia. We cover all, we just do it all at once. So we cover more, we humans have more than 25,000 genes, and you just see the proportion, single gene, more than 120 genes, and 25,000 genes. And you see really how the power increases to cover the genes. And interestingly, this is even more cost efficient than the panel approach. This just costs 1,200 euro. Now it's up, maybe in some centers it just costs 900 euro. And it just takes 3 months. So this is even more cost efficient and even more time efficient than the panel approach. So why do we not just start with the exome in all of our patients right away? Why do we have to care about this at all, about the panel approach? The problem with the exome approach is that although it does cover all 25,000 genes, it has holes. It does not cover all places of the genes very well. There might be some holes, so some parts of the genes might not very well covered, and those holes are there at all. It's like a patchy network. There are always some holes in our coverage. And what you then have to do is go to those holes and cover them, and do them by conventional Sanger sequencing. So the sensitivity is a little bit lower of the exome sequencing, and this is why we're not doing it right away. Whereas in the panels, those panels are specifically profiled to cover all genes with the so-called high coverage. So there are no holes, it's no patchy network, you just cover them all very well. So it has an excellent sensitivity, better than exome sequencing. Now I want to give you 2 examples of how we can use this in practice to find a genetic diagnosis for patients where so far we had no idea what the genetic diagnosis is. And I'm going to give you 1 example for panel sequencing and 1 example for exome sequencing. Let me start with the example for panel sequencing. Just to remind you, where do we use panel sequencing? Panel sequencing we use to cover all known ataxia genes. And just to make it more illustrative, to cover this large share of recessive ataxias, which we cannot do one by one by this Sanger sequencing. This will take us years and hundreds of thousands of euro if we do it by Sanger sequencing. So what we do, we just cover the whole set by a panel. And how, just give you an example from practice. We had a boy, and you will see this later on again. This boy had a slowly progressive cerebellar ataxia, but no additional symptom. And with such a boy, you're pretty much lost in clinical neurology. If you do not have any additional symptoms where you raised alpha-fetoproteinosone, if you do not have any additional feature, you have pretty much no idea where you should look for. This is not Friedreich's, it's purely cerebellar. Friedreich's is not cerebellar, and at least not purely cerebellar. So you can now start to go gene by gene, go for your Sanger sequencing approach, and this will take you forever. There are at least more than 50 genes out there which might cause a purely slowly progressive cerebellar syndrome. So this standard approach will not work. You cannot cover by Sanger sequencing gene by gene by gene. So you need to do this panel approach and cover all the genes. You really go for all 100 genes at once, 120 genes at once, shoot at all of them at once. And in fact, we were successful, and we're successful in finding mutations in a gene called STAP1. You don't have to remember the names. This is a problem. You can't remember all those 120 names. But just to give you a glimpse how successful this approach is. So we found the mutations in a gene called STAP1, and this gene has just been identified in 2014 by a group before us, and it's known to cause ataxia syndrome, so cerebellar atrophy combined with hypogonadism, with reduced sex hormones. And this boy, interestingly, did not have sex hormones, so we would reduce sex hormones. He did not have hypogonadism, so we would in the first stage we would never have thought to go for this gene. We would never have thought to sequence STAP1 because this gene, this boy just had slowly progressive cerebellar ataxia. So we would never have gone for this gene. We just found it so along the route, by the way, by shooting at all of the genes at once. So what does it help us? We can now provide this boy with a genetic diagnosis. And we're now trying to find out what's the functional role of STAP1. And it seems to be really is a crucial protein in ataxia pathways. And once there's going to be a therapy, this boy will be included in treatment studies in the future. Without having a diagnosis, this boy could not go into individualized treatment studies in the future. So this was an example for panel sequencing, where you cover all known ataxia genes. Now I'm going to give you an example of whole exome sequencing. What is the target of whole exome sequencing? With whole exome sequencing, you also cover those known genes, of course, but the main advantage is that you're able to find genes, even in those patients which do not have a mutation in any of the known genes, so which have a mutation in a new gene. And how does whole exome sequencing work? You sequence all 25,000 genes, and you have to filter then in order to find the really crucial, essential gene variant which is causing the disease in your patient. And I want to give you an example of a patient where we use this approach. This young woman had ataxia, but in addition, vision problems and reduced sex hormones. So she did also have hypogonadism. And she always asked me, is this all due to one ataxia disease? Or do I have 3 or 4 diseases? And I said, I have no idea. I presume that this is one idea, but I have no idea what the cause is. And she kept coming to me each year, and we had no idea what this was. So, and she had even some cognitive deficits, some spasticity in her legs. So all of this seemed to be one syndrome, but we had no idea what this was. In the literature, this syndrome is called Buschet-Neuhauser syndrome. Cerebellar atrophy, retinal dystrophy, and hypogonadism. This triad is called Buschet-Neuhauser syndrome. So this is how neurology worked in the past century. We had nice terms for our diseases, especially we neurologists are really good at giving complex syndrome names, especially like Synofzik syndrome or whatsoever. But this doesn't explain anything. It's not helpful at all. So and nowadays we really want to find the molecular cause of our disease. And I could tell the patient, okay, you have Buschet-Neuhauser syndrome, but so what? She will not care about this. She wants to know the real cause of her disease. So what we did is, this woman had a sister with the same syndrome. So we did an exome of both of them. And exome sequencing works like this. In exome sequencing, not the sequencing per se is an interesting step. That is trivial. Any second commercial genetic center now does exome sequencing. That's not the crucial step. The crucial step is that you have to use filters in order to filter down all of 25,000 genes in order to find out what is the main gene. And this is just an illustration that you filter out only for those sequences which are rare, which are not in control databases. And you only want to filter out those variants which are predicted to be damaging. And thus you filter out and try to reduce your long list of gene variants. But very often in our Western European world, you still come up with 5 to 50 candidate variants in the end. So meaning very often there's still quite a few number of variants which we end up with by exome sequencing. And then the problem is we do not know which of those 5 to 50 gene candidates really is now the true cause of the disease. So if you have large families with several affected persons, 5 or 6, you can look, okay, do they share a gene variant? You can do an exome of all of them and look, are there shared variants in those 5 to 6 siblings? And then this would be the crucial one. But in our Western European world, we have very often just very small families, and if at all, 2 siblings. And then you end up still with 5 to 50 candidate variants. So what we did in this family, we put their exomes of those two sisters in a large exome database. And that was a focus on one of the talks last Wednesday, that the need for large exome databases, that you integrate your data together. And in this exome database, we found 5 other families with the same phenotype: hypogonadism, ataxia, and retinal dystrophy. And all of those 5 families had a variant in the very same gene, and the gene is called PNPLA6. Again, you do not have to remind of this particular gene name. It's just it's a principle which is important here, that you need to have large families or large exome databases, type in the phenotype, and this will allow you to find whether certain families share the same gene variants and allows you to provide a genetic diagnosis. And PNPLA6 is, for example, is a protein which is involved in phospholipid metabolism, and there's now an increasing number of spastic paraplegics and ataxias where we do know that they have a problem phospholipid metabolism. So this seems to be a common hub in ataxias and HSPs, and maybe we are going to develop therapies for this common hub, which we would not have done if we would not have known that this is a crucial gene in this pathway. So this is where it helps. It helps us for the individual patient to provide a diagnosis, and it helps us to identify common pathways shared by different ataxias and now make this target of our future therapies. So this was an example of exome sequencing. I've been giving you an example of panel sequencing and an example of exome sequencing. And this is actually all I wanted to point you out, that you just get an idea about panel sequencing and exome sequencing, because you will find those terms in the main conference. And everybody of us is using either one or both approaches, and it's really now successful in providing genetic diagnosis to patients and in given exomes. So this is the main part of my talk, and I'm pretty much done. But just Julie and me, we thought there's a new... Yeah, this is perfect. There's a second study which has not to do at all with exomes or panels, but which is supported by UK Ataxia, and we really love this study, and many of our patients love it, and that's the reason why I just make a small 2 slides spent on 2 studies which we have done beyond genetics. And in those studies, we were asking whether Exergames, you can use Exergames for rehabilitation as a treatment tool in ataxia. What are Exergames? Exergames are video games which you play with your whole body. Not just with your hand, you're not just playing around with a Game Boy or something, you're playing with your whole body, with your arms, with your legs, with your whole body, and you're active. And this is used to use, for example, the Microsoft Xbox or Nintendo Wii gaming console, and you play with your whole body. And the question we were asking is, can you use this as a therapy to treat ataxia? So play in a gameful, motivational play way and reduce ataxia. And this is a boy, you might remember this boy from earlier, this was the boy with the STAB1 mutations. And this boy is playing table tennis. And we're trying for table tennis was one of the games we wanted to find out, does this improve ataxia? And you see the game forces the boy to go out of his center of mass and do movements far away from his center of mass, and to really, and it goes directly into his ataxia deficit, meaning bimanual coordination and legs-arm coordination. So do those games help? And we have done a study in 2012 where we're able to show that those Xbox games do help the patients to improve their ataxia in a playful way. This was the first study which was supported by Ataxia UK, and because those results were so promising, we did a second study, and we went 1 step ahead and said, okay, if they are successful, effective in subjects who are still able to walk and to stand, could we also use them in those subjects who are not able to walk and stand anymore? Can we use them in wheelchair-based subjects? Because wheelchair-based subjects, very often there's no therapy and they're excluded from common trials. If you look at current free trial trials, wheelchair-based subjects are currently excluded from those trials. So there's not much you can offer them. Now, can we offer them exergame training? And the idea which we had is we use the V platform, but we take it off-label. The V platform was created to stand on it. We say, okay, this is not helpful. This will not work in our patients, which you're focusing on. We just have patients sit on the Wii platform and control the Wii platform by sitting. So we use the Wii platform off-label. I'll just give you an example of a child here. So the child has to relearn trunk control, and she's having fun. And she has to balance a board and try to get a ball into the hole. But the ball should not fall off the board on the side, but it has to go in the hole. So she has to really try to regain trunk control. And if patients, so we had 2 phases in the study. The first one was sitting, just to regain trunk control. And if they were able to regain trunk control, we had the second phase where we tried to make them stand, if they could still stand somewhat, and try to regain at least some stance control. So then we had them stand on the Wii platform and play the same or other more complex games while standing. So this is this is a study we are now finishing off, and we have now 10 patients and preparing this for publication. I think we have to take 1 to 2 more patients, and the first results are really promising. We have a poster out there in the poster hall, and thanks to Ataxia UK. And this Exergames is the studies are so much fun, and they're really to the patients, and they're really efficient, that we're now embarking on a third study where we want to check whether you can use Exergames, tablet games, and so on for training upper limb for training upper limb coordination. And this is a new proposal for UK Ataxia, whether you can use upper limb Exergames to improve upper limb control. So to sum up, what I've shown you is that Friedreich is by far the most common recessive ataxia. This was those 25%. And this is why it's such a major focus here on the conference. But it's important for you that there are many, many other recessive ataxias out there. More than 100 other recessive ataxias, and we should not forget about them. As a whole, they are also 25%. Not only each one is less, but as a whole, they also account for as much as Friedreich's ataxia. And what we can now do is screen all of these 100 other recessive ataxias, not by one, but screen all of them by this panel approach. And this is time efficient and at relatively low cost. And for all the patients who do not have a gene mutation in a known gene, we have a second approach. We have an approach called exome sequencing, where you shot at all genes at once. And by the panel sequencing, we are currently able to provide another 25% of patients with a diagnosis. So I think this should become a new standard of care in the future for all patients with recessive ataxias, where you do not have a straightforward guess that it's Friedreich ataxia or AOA2. If you do not have a straightforward guess, go for a panel sequencing. And if you're not successful with panel sequencing, go for exome, or in the future maybe even genome. And as I've just shown you for the young woman with the retinal dystrophy, this provides several families with a diagnosis and now a clear cause of the disease. So we no longer deal with those strange terms like Busché-Neuhauser syndrome. We can now provide a molecular genetic diagnosis to our patients, for example, using exome. And taken together, the panel sequencing and the exome approach, I think already quite a few numbers has allowed us to stop the diagnostic odyssey for many patients. Those patients have been there for many, many years. And they go to the doctors and do not find a diagnosis. And panel and exome help to make the diagnosis. And, which is of particular importance, if you have a molecular diagnosis, this makes our patients accessible for disease-specific treatment studies. As I said, molecular diagnosis is the first step for molecular therapy. Without molecular diagnosis, there will not be a good molecular therapy. And we're now putting together an application for a large European grant, the so-called PREPARE initiative. And we are now collecting patients with all kinds of rare recessive ataxia. So all recessive ataxias except Friedreich. And we're compiling them in a registry. And for 2 recessive ataxias, RSAX and ADCK3, we really want to make them ready for trial. And we want to compile cohorts of patients with rare ataxia, so as soon as there's any other drug for rare ataxia, that we are ready, that we have cohorts large enough to let them go to trial. Because if there's a pharma company which has a drug and we have to start to screen patients, look for patients, this will take forever. We want to be ready for trials, and this is why we started this initiative and started a large registry. Thank you very much for your attention.